न्यूक्लिक, अब देखो एवरी सेल प्लांट सेल हो या एनिमल सेल हो, देयर विल बी न्यूक्लियस, और न्यूक्लियस के अंदर क्रोमोसोम्स होता है, ठीक है। एंड दीज क्रोमोसोम्स आर मेड अप ऑफ प्रोटीन्स, ये याद रखना, क्रोमोसोम्स आर मेड अप ऑफ प्रोटीन्स एंड न्यूक्लिक एसिड, ठीक है। क्रोमोसोम्स के अंदर होता है, वी आर गोइंग टू टॉक अबाउट व्हाट आर क्रोमोसोम्स मेड अप ऑफ। क्रोमोसोम्स आर मेड अप ऑफ प्रोटीन्स एंड न्यूक्लिक एसिड्स, ठीक है। और न्यूक्लिक एसिड, देयर आर टू टाइप्स ऑफ न्यूक्लिक एसिड, DNA and RNA. Write down DNA again. Revision. Okay. So there are two types of nucleic acids, DNA and RNA. Okay. Write down the full form of DNA, deoxyribonucleic acid, and RNA is ribonucleic acids. Okay. Write down. Nucleic acids are also known as polynucleotides. Very important. Nucleotide. If you remember, tide used to wash clothes. Tide. Tide. Okay. Polynucleotides. Nucleic acids, they are also known as polynucleotides. We'll see what are these nucleotides. There is something known as nucleoside also. There are two terms, nucleotide and nucleoside. So nucleic acids are polynucleotides. Write down. Nucleic acids are polynucleotides. So this guy, James. What's the difference between amino acids and okay? What's the difference between amino acids and proteins? Huh? Yeah, check your notes. What's the difference between amino acids and proteins? Alpha amino acids and proteins. What's the difference? Who can tell me? Shupriyata, what's the difference between alpha amino acids and correct? So one unit is known as alpha amino acid. Many of them are known as proteins, right? It's the same thing. जैसे कि one nucleotide is, we'll see what is a nucleotide. Many of the nucleotides are known as nucleic acids या polynucleotides, right? ठीक है, फिर से मैं बोल रहा हूं, we are talking about nucleic acids. So nucleic acids are found inside the chromosomes. Chromosomes are made up of two things, proteins and nucleic acids. ठीक है, nucleic acid के दो टाइप होते हैं, ribonucleic acid, RNA, and DNA, deoxyribonucleic acid. And there are two terms, two new terms, nucleotide, nucleoside. Okay, nucleotide and nucleoside. So nucleic acids are polynucleotides, not nucleosides. I will explain what are these two terms right now. Write down, nucleic acids are polynucleotides. And what is a nucleic acid made up of? If you take DNA, guys, what's the difference between nucleotide and DNA? Are both these two things same? What is DNA? Because DNA nucleotide. What is the difference between insulin and glycine? नहीं, glycine नहीं, lots of something. Insulin is a polymer. It will have many alpha amino acids. Glycine बस एक. Same चीज यहाँ पे. DNA, RNA are polymers of nucleotides. But a nucleotide is the individual unit. Understood? There is a difference between DNA and a nucleotide. DNA is made up of many nucleotides. ठीक है? DNA is a polymer. Right. Okay. So then we are going to talk about the structure of DNA. So इस बंदे ने ये स्ट्रक्चर निकाला था, लाइक पूरा रिवोल्यूशनरी डिस्कवरी of the structure of DNA. ठीक है? So the structure of DNA is something like this. I mean, it's like this. And see properly, you will understand what I'm doing. I'm writing some numbers. You'll understand those numbers. So ये जो okay, so let's say you have a ladder, right? So what are these known as? What are these known as? The side के जो उसका एक नाम है, like they have given one name here, rails. These are known as the rails of the ladder. The side one. And these which are connecting the two ladders, these are known as rungs. ऐसा कुछ नाम है, क्या है? Rungs. Okay, so these are your rungs. DNA का स्ट्रक्चर भी कुछ ऐसा ही है. This is the rail. Are you able to see that? This is the rail. This is the rail. ठीक है? और यहाँ पे, like I told you, if you hydrolyze a DNA molecule, what you are going to get? Three things. Phosphoric acid, H3PO4. दूसरा प्रोटीन मिलेगा इसमें. न्यूक्लियोटाइड, हाँ टेक्निकली सही है, लेकिन सबसे देखो न्यूक्लियोटाइड इज, आई मीन सबसे बेसिक चीज बोलो ना. न्यूक्लियोटाइड में गया ही नहीं. आई टोल्ड यू देयर यू विल गेट थ्री थिंग्स व्हेन यू डू हाइड्रोलिसिस ऑफ ए डीएनए molecule: sugar, base, and phosphoric acid. Come on, check everyone. Check everyone. I have told you. Have you written in your notes? Have you written? Did you write? I told you this. How many times I repeated this in the last class? Whenever you do hydrolysis of a DNA molecule, you will get three things: phosphoric acid, H3PO4, then you will get a sugar, and then you will get bases. Bases are heterocyclic compounds containing nitrogen. This is not our regular base. Okay? So DNA molecule में हम लोगों ने ये ऑब्जर्व किया है कि फर्स्ट ऑफ ऑल कि these rails, ये जो imagine this as the ladder. So these rails are made up of sugar and phosphoric acid. ठीक है? और the rails of these, ये समझ में आ गई, ये एक चेन है. समझ में आ रहा है? जैसे पॉलीपेप्टाइड चेन था, ये एक चेन है. आई विल यूज अनदर कलर. ये एक चेन है. डीएनए जो मॉलिक्यूल होता है ना, दैट कंसिस्ट ऑफ टू स्ट्रैंड्स. आर यू गेटिंग व्हाट आई एम सेइंग? हर एक डीएनए मॉलिक्यूल में दो चेन होता है which combine together to form a alpha helix. Do you remember? पॉलीपेप्टाइड चेन में मैंने बोला था सेकेंडरी स्ट्रक्चर याद आया? अल्फा हेलिक्स. दो दो चेन ऐसे. द सेम चीज डीएनए में दो स्ट्रैंड होता है, याद रखना. इट कंसिस्ट ऑफ टू सच चेन्स ऑफ न्यूक्लियोटाइड्स. ठीक है? ऐसे चेन होगा. एंड इस चेन में यू विल हैव शुगर. ये एक चेन है, ये दूसरा चेन. सो इस चेन में यू विल हैव शुगर एंड फॉस्फोरिक एसिड, एंड these two chains are connected by the rungs. See the rungs. दो सीढ़ी के जो दो एंड को who is connecting? The rungs. The rungs are the bases. एक बेस यहाँ पे होगा, मान लो यहाँ पे एडिनाइन है, यहाँ पे थाइमिन होगा. चेक करो एडिनाइन और थाइमिन क्या है. ग्वाइनाइन और साइटोसिन क्या है? चेक करो. These are the four bases that are found in DNA. So एडिनाइन और थाइमिन के बीच हाइड्रोजन बॉन्डिंग बने. ग्वाइनाइन और साइटोसिन के बीच हाइड्रोजन बॉन्डिंग बने. So, are you getting how these chains are connected to each other? There will be two chains, and the chain is formed because of sugar and phosphoric acid. But hydrogen bonding between the bases connects them. So that's the structure of DNA. These are the name of the bases. T is thymine, C is cytosine. A is adenine, G is guanine. Okay. RNA is completely different. RNA is a single strand. ऐसा नहीं है, RNA ऐसा नहीं है दो, RNA ऐसा होगा, एक ही strand. Okay. Fine. So, like, explain this in your own words, a bit of it. You will understand it better, or be better understanding. That's the numbering in the chain that I will explain in a while. I mean, you'll understand it better. I will show you some diagrams. See this diagram? Are you able to see this diagram? So S is your sugar, P is your phosphoric acid, and ये जो देखो ये T और A क्या है? Basis. So this end is formed by the sugar and the phosphoric acid group, and these two chains are connected by hydrogen bonds between these bases. Check everyone. Adenine और thymine के बीच ही bond होता है. I mean, a bond cannot happen between adenine and guanine. And everyone, I mean, both of you look here, all three of you. Let's say one strand is like this, okay? And you want adenine and thymine here, adenine and thymine. Can the strands be similar? अगर मान लो यहाँ एडेनाइन है, यहाँ एडेनाइन, एडेनाइन एडेनाइन बॉन्ड नहीं हो सकता, इसीलिए उल्टा होता है. एक चेन अगर ऐसा हो रहा है ना, दूसरा चेन ऐसा हो, इसीलिए उसका 3 डैश और 5 डैश से जा रहा है, इसका 5 डैश से 3 डैश जा रहा है. Both the chains are complementary. Are you getting what I'm saying? Because like the molecules are like that. Hydrogen bonding, I mean, it's like that. Adenine cannot form a bond with guanine. It is like that. और एक रीजन ये भी है ना कि guanine और cytosine जो है, they have 3 hydrogens. They can form 3 hydrogen bonds. How will adenine form a bond with guanine? Because adenine at max can form 2 hydrogen bonds. तो adenine और thymine का होना जरूरी है. Are you getting? Makes sense. That's why both the strands are opposite. Are you able to see that? That's why the number, you'll understand it better. Just draw this diagram roughly. These are individual nucleotides. Individual nucleotides. एक पूरा चेन को क्या बोलेंगे? Polynucleotide. Keep your video on, Shupriyata. Are you there? Okay, so now we will explore the structure in detail. Okay, so write down these points. Complete hydrolysis of DNA or RNA will give you pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compounds, which are known as bases. Write down, very important. What will be the hydrolysis of polysaccharides give you? Monosaccharides milega na? Similarly. These are polynucleotides. So you will get nucleotides. And DNA में कौन सा sugar है? देखो, in this figure, I mean, if you can see here in this figure, you can see S. You can see that S. What is that? That's sugar. Okay, and I'm going to mention what is that sugar. Okay, so the sugar is in DNA it is beta D2 deoxyribose, and in RNA it is beta D ribose. Draw their structures. ठीक है। In DNA, the sugar is beta 2 deoxyribose. This is your sugar. और RNA में sugar होता है beta D ribose. This is the sugar. Correct? Sugar is carbohydrate, right? Okay, carbohydrates are hemiacetal या acetal. Acetal, right? कि hemiacetal. Hemiacetal ना, because एक molecule react कर. So we are breaking down the structure. So we will see what's the sugar first. So DNA में ये वाला sugar होता है. Sorry, DNA में ये वाला sugar. This is your sugar in DNA. And draw the structure of H3PO4 also, phosphoric acid. That's P double bond O, OH, OH, and OH. This is the structure of phosphoric acid. This is the structure of phosphoric acid. This is the only difference. यहाँ पे H है, यहाँ पे OH. एक H, एक ऑक्सीजन एटम की वजह से देखो क्या चेंजेस आते हैं, right? So understood till here. So we understood the sugar, right? Now we understood the phosphoric acid. Now we are going to talk about the bases. Bases में फिर से मैं बोल रहा हूँ, what are bases? These are not the conventional bases that we have been studying till now. These bases are heterocyclic. Heterocyclic मतलब कि cyclic है. Hetero मतलब होता है a atom which is different from carbon. Cyclic है और कार्बन से हटके, I mean कार्बन से एक अलग एटम चाहिए. Okay, and so that atom is nitrogen here. So this is cyclic, but in the ring you have nitrogen. So these are all heterocyclic compounds. So you have 4 such bases in DNA: adenine, guanine, thymine, guanine, cytosine. ऐसे याद रखना, ATGC, because A and T are connected. Similarly, T and A are connected. Similarly, G and C are connected. C and G are connected. ठीक है, ऐसे याद रखना, बेसिस के बीच कनेक्शन होता है. Always remember. मैंने क्या बोला था? The shape of DNA is like your ladder. So these are the chains of your sugar and phosphoric acid, and ladder is connected by the bases. तो bases के बीच connection होने के लिए you should have A and T, T and A. Okay, so this is your adenine, this is your guanine, this is your cytosine, this is your thymine. But एक issue ये है कि RNA में क्या होता है? Adenine होगा, guanine होगा, cytosine होगा, thymine नहीं होगा. Instead of thymine you have uracil. Don't ask me why. It is what it is. And draw their structures. Adenine, guanine, cytosine, thymine and uracil. ठीक है, अभी अभी मत बनाओ, अभी बाद में बनाना. बट यू शुड अंडरस्टैंड दैट दिस इज व्हेन आई राइट ए, ओके, व्हेन दीस गाइज वर राइटिंग ATGC, they mean these compounds, but I don't want to draw that every time. ठीक है, okay. And if you see adenine and thymine, ध्यान से देखो, adenine में how many hydrogen bonds you can form? How many hydrogens are freely connected to nitrogen? Adenine में 3 है क्या? And in thymine, में कितना है? 2. और guanine में कितना है? 3. साइटोसिन में कितना है? 3. ग्वानिन हैज 2 3 4. ओके ओके ओके. एडिनाइन और थाइमिन के बीच बॉन्ड होता है ना. हाँ, ओ और ओके. ग्वानिन और साइटोसिन के बीच बॉन्ड होता है. ठीक है, तो 2 और 1 3. एक सी एडिनाइन में अंडरस्टूड. यहाँ पे दो हाइड्रोजन एटम. यहाँ पे 2 हाइड्रोजन एटम है. यहाँ पे सीओ है, सीओ है. तो 2 यहाँ पे हाइड्रोजन बॉन्ड मिल गया है. अबाउट ग्वानिन एंड साइटोसिन. ग्वानिन एंड साइटोसिन में सोच के देखो, हाउ इज इट पॉसिबल दैट दे फॉर्म 3 हाइड्रोजन बॉन्ड्स. लोड लोड लोड. लोड क्यों नहीं हो रहा है? Yeah, are you able to see this? Are you able to see thymine and adenine? They are forming 2 hydrogen bonds and 3 hydrogen bonds in guanine and cytosine. Hydrogen bond with night. Yes, एक highly electronegative atom होना चाहिए. In adenine and thymine, none of the oxygen. Why? यहाँ पे बंद हो रहा है. देखो थाइमिन का ऑक्सीजन एंड. हाँ, ठीक है. ये वाला चीज देखना पड़ेगा. बिकॉज मोस्ट हर जगह पे मोस्ट ऑफ द टाइम दे से दैट एडेनाइन एंड थाइमिन, साइटोसिन एंड ग्वानिन ऐसे ही बोलते हैं. बट दे डोंट एक्सप्लेन कि दिस इज व्हाट आई फाउंड इन द. दिस एक्सप्लेन्स द 2 हाइड्रोजन बॉन्ड बिटवीन द 3 हाइड्रोजन बॉन्ड्स. ठीक है, ये मैं बाद में बताता हूँ. ओके फाइन. सो नेक्स्ट राइट डाउन न्यूक्लियोसाइड. Nucleoside. Now we will understand what is a nucleoside and what is a nucleotide and what's the difference between them. So to understand what's a nucleoside, first draw a sugar. Draw beta D ribose. They say what is this sugar? Yeah, they have drawn beta D ribose. Draw beta D ribose. ठीक है, draw beta D ribose. Yeah, जो RNA का sugar. Or you can draw 2 deoxy ribose also. Draw a sugar. See, basically what I want to say is कि ये sugar है मान लो. Okay, see here everyone. This is your sugar in RNA. This is your sugar in DNA. ठीक है. So first carbon पे, guys focus. First carbon पे, this is your first carbon, right? अगर इस carbon पे you have connected a base. I mean, एक base has formed a bond with this carbon. Are you getting what I'm saying? Sugar है. देखो, whenever you do the hydrolysis of polysaccharides, right? What is the bond connecting the individual monosaccharides? What is that bond known as? Glycosidic linkage. Similarly, यहाँ पे, whenever you are doing hydrolysis of DNA, you are getting 3 things: sugar, base, and phosphoric acid. So something must be connecting them, right? Okay. So we have observed that you have sugar, and in the first carbon, it is connected to a base. Then this is known as your nucleoside. Is this nucleotide? नहीं है ना, क्योंकि फॉस्फोरिक एसिड तुमने लगाया ही नहीं है अभी तक. Are you getting? The moment I attach one phosphoric acid, then it becomes a nucleotide. Are you getting? शुगर है, शुगर में मैंने बेस लगा दिया. So then it is nucleoside. But the moment I add one phosphoric acid also, then it becomes a nucleotide. That's not how we define. Okay, we define it in this in such way. Good question. शुगर में जैसे ही बेस ऐड कर दिया, then it becomes nucleoside. Okay. So similarly ये देखो, ये जो शुगर था, देखो फर्स्ट एवरीवन गाइस, दिस इज बीटा डी राइबोस. ठीक है, उसमें फर्स्ट पोजीशन पे बेस लगा दिया. एवरीथिंग एल्स इज सेम. सो देन दिस इज अ न्यूक्लियोसाइड. ठीक है, एंड देन यू सी. ठीक है, ये फिर से शुगर है. विवान, ये फिर से शुगर है. ओके, आई अटैच ए बेस एंड देन आई ऑल्सो अटैच्ड अ phosphate group. Understood? So then it becomes a nucleotide. That's the difference between. हाँ हाँ, जोर से बोलो. क्या बोल रहे हो? Phosphoric acid, it can lose its hydrogen. Huh? I mean, if it is in a aqueous medium, तो lose कर सकता है ना as a proton. ठीक है? You can देखो. That's why there is a negative sign here. If you want to keep H, you can keep H. Okay? देखो कैसे बॉन्ड बना गाइस, ऑब्जर्व. यहाँ पे CH2OH था और यहाँ पे you have H2H3PO4, राइट? तो यहाँ पे एक OH निकल गया, यहाँ से एक H निकल गया. So you got a bond here. Understood? Understand? Are you able to understand how to draw a nucleotide? न्यूक्लियोसाइड में क्या करना है, शुगर बनाना है और फर्स्ट पोजीशन पे एक बेस लगा देना है. खत्म. न्यूक्लियोटाइड कैसे बनाना है? लाइक CH2OH है, इसमें से OH निकाल दो और H3PO4 में से H निकाल दो. So you will get a nucleotide. ठीक है. अभी अब दिस इज अ इंडिविजुअल न्यूक्लियोटाइड. what are nucleic acids? Polynucleotides, right? So understood what is a nucleotide, right? अभी हमें देखना है how will you get a polynucleotide. ठीक है. So first of all you need to draw a nucleotide. So this is your nucleotide. एक nucleotide, we will draw a nucleotide. ठीक है. O. See these numbers, 5 dash or 3 dash. Your doubt will be cleared. No issue. Okay, draw one more. Draw one more nucleotide. Okay, so these are the different nucleotides. Are you able to see that, guys? Thus, this is one nucleotide. This is another nucleotide. Okay. Now see the numbering carefully. This is one dash, two dash, three dash, four dash, five dash. This is again one dash, two dash, three dash, four dash, five dash. Okay. तो ये दोनों अलग अलग न्यूक्लियोटाइड है, right? Why I am giving dash to these numbers is because बेस में द बेस, व्हाट इज बेस? बेस भी याद होगा ना, ऐसे नाइट्रोजन का एक साइक्लिक कंपाउंड है, बेस में डबल बॉन्ड है. In the base, I have already given 1, 2, 3, 4, 5, 6 numbering. That's why I'm using dash. Have you understood what I'm saying? Nomenclature में कर रहा हूँ ना, तो if I have used 1, 2, 3, 4 in one ring, दूसरे ring में I have to use 1 dash, 2 dash, 3 dash, 4 dash. That's the reason. That's why I'm using dash. ठीक है? Okay. Now ये ध्यान से देखना. अभी ये देखो कि 5 dash, अब now see the 3 dash end. 3 dash end में OH है, और दूसरे न्यूक्लियोटाइड में 5 dash end में एक, like you have CH2 and this is your phosphate group, isn't it, guys? Right? Now this bond, now what will happen? I mean, how, if you remember, दो ग्लूकोज के बीच कौन सा बॉन्ड था? ग्लाइकोसिडिक लिंकेज. ठीक है. यहां पे दो न्यूक्लियोटाइड के बीच जो बॉन्ड होगा, दैट इज नोन एज फॉस्फोडाइएस्टर बॉन्ड. ठीक है, देखना अभी कैसे होगा वो. हाँ, फॉस्फोडाइएस्टर. ठीक है, अभी फर्स्ट ऑब्जर्व सी व्हाट विल हैपन. यहां से एक OH निकलेगा, यहां से एक H निकलेगा. ओके, सो यू विल गेट these two nucleotides are connected. Okay, similarly same thing will happen here also. Are you getting what is happening? देखो, so you started from the 5 dash end. 5 dash end free है, and then 3 dash end में क्या हो रहा है? Like you're going. Ultimately, which end will be free in the end of the ring? See, if you started from 5 dash free है, and if let's say the I mean the bond is forming between two nucleotides, मान लो एकदम लास्ट में पहुंच गए, the 3 dash end will be free, right? Because और एक नहीं है. Right here the 3 dash end is not free because यहाँ पे एक है और एक है, दोनों एक बॉन्ड बना. See, this guy is 3 dash and this guy is 5 dash is making a bond. Similarly, this guy is 3 dash and this guy is 5 dash is making a bond. In the end, the 3 dash will be free, no? In the end of the chain, because there is no other nucleotide. So that's what I meant when I showed you this 5 dash to 3 dash, 3 dash end. और यहाँ पे 3 dash to 5 dash उल्टा होता है. That's what is the meaning of this 5 dash to 3 dash, 3 dash to 5 dash. I hope you understood. ठीक है. See here. See this. शुगर है. हाँ, नहीं, लाइक पहले वाले का 5 डैश इज फ्री, लास्ट वन 3 डैश इज. हाँ, क्या 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 क्या. ऐसा नहीं होता ना, लाइक बोथ आर डिफरेंट चेन्स. दिस हैपेंस इन RNA, दिस डज नॉट हैपन इन DNA. DNA में दो स्ट्रैंड्स आते हैं, लाइक एक 5 सेम थिंग, एक का 5 डैश से 3 डैश जा रहा है, एक का 3 डैश से 5 डैश. बिकॉज वो एडेनाइन और थाइमिन के साथ बॉन्ड बनने से ना इट बिकम्स मोर स्टेबल. So that's the thing. So are you able to see here? This is your sugar. This is your base. 3 dash 5 dash 3 dash 5 dash 3 dash 5 dash ऐसे चलता है. Okay. Understood? Okay. So this is what happens. See here, guanine, cytosine, this triple bond means three hydrogen bonds, not triple bond. Adenine, thymine, thymine, adenine, adenine, thymine. And this जो ये basis का जो order है, that's unique in every human. जो basis का order है, जैसे तुम्हारा क्या हो सकता है, adenine, thymine, adenine, thymine, 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 guanine, cytosine. मेरा अलग होगा. That's what is known as DNA fingerprinting. That's what they check. When like CID वगैरह में देखा होगा, they always collect the DNA of the criminal, if you remember. What do they do with the DNA? They check the basis, the order of the basis. That's unique for everyone. सबके लिए unique. Understood? What is actually going on? To verify, the person, like what we'll do is, let's say XYZ ka le liya, let's say he is the criminal. And then we will collect the DNA of all the suspects. And then we will match it. कि यही बंदा है क्या? The way you do fingerprints, right? Fingerprints करके क्या करोगे? You'll be a very bad detective. Okay? So, और DNA ऐसे लिखना, DNA का क्या होता है? Single stranded helix. Write down DNA, single stranded helix. मतलब ये देखो, single stranded, बस एक strand. Sorry, RNA. And there are 4 types of RNA. Write down: messenger RNA, ribosomal RNA, transfer RNA. 3 types के हैं क्या? 3 types. Sorry, there are 3 types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. DNA fingerprinting, ठीक है. And like पेरेंट्स से जो दूसरा जो नेक्स्ट जनरेशन होता है, डीएनए पास होता है. ठीक है, डीएनए पास होता है. एंड रीड हार्मोन्स योरसेल्फ. कुछ भी नहीं है. ठीक है, अभी बढ़ो. डिड नॉट, ये बोला है, उससे पढ़ लो. रीड हार्मोन्स योरसेल्फ. हार्मोन्स ऐसा कुछ नहीं है. ऐसा नहीं है कि कार्बोहाइड्रेट ही होगा या प्रोटीन भी होगा. हार्मोन्स आर मॉलिक्यूल्स विद अ स्पेसिफिक वर्ड डिजाइन टू. हार्मोन्स कैन बी प्रोटीन्स. ठीक है, insulin is a hormone, but insulin is also a protein. So hormone is based on the function that they do. Okay. एग्जाम्पल देख लेना, टाइप्स देख लेना, इसमें और कुछ नहीं है. हॉर्मोन्स में ऐसा कुछ नहीं है कि उसका केमिस्ट्री पढ़ना है, कुछ भी नहीं है. उसका एग्जाम्पल है, उसका यूज है, देख लेना. ओके. एंड देन रिवाइज द होल चैप्टर of this. Yeah, you want to do, then like do one thing, everyone. After you are done, first of all read what are hormones, then read the summary. Okay, then start the NCRT. Insulated insulin. People think of insulin. Okay. Yeah, that I know. To, इंसुलिन का काम क्या होता है? To decrease the glucose levels, right? Okay. Insulin is a protein. Protein can be man-made. Maybe they discovered how to make insulin. Protein ही तो है. Steroids are also made now. Steroids, so you must be knowing. Those are used by sports persons, right? Do you know that? Paul Pogba, what happened to him? Huh? Yeah, like, that's why they do drug tests, right? To check these chemicals, enhanced chemicals. Why? No, but it's unfair to other people. Why? Why are you disturbing your bodily composition? Then you are justifying the use. नहीं, like, see, these are first of all पैसा लगता है, सबके पास थोड़ी ना है. Then how is it equal for everyone? वाह, and it's not also good for your body. Foreign materials. दांत में मेरे गए भाई. क्या हो? Hmm? Yeah, that's the. Hmm. Torali, Dulin, Shaman, keep your video on. Hmm. Hmm. हो गया. Done with reading hormones. ठीक है. अभी समरी पढ़ लिया. Write down the summary. Start writing the summary. इससे देखो ना, मैं थोड़ा काम कर रहा हूँ. Start writing the summary. वो कुछ फैट सोल्यूबल या वाटर सोल्यूबल है, वो फिर रिलीज हो जाता है। Check what is that. Vitamin C is water soluble or not? Check. विटामिन सी वाटर सोल्यूबल होना चाहिए, देखो। विटामिन पे जाओ, वाटर सोल्यूबल है ना, सो इट गोज आउट इन द फॉर्म ऑफ यूरिन और वट एवर, लाइक पर्सपिरेशन, स्वेट से भी जा सकता है, तो इसीलिए कंटिन्यूअस इनटेक चाहिए उसका। Next class में, like those who wants to read P block, it's not there in your boards. P block किसको किसको पढ़ना है?
Sir, can you revise coordination compound? One plus.
Revise.
Yeah. One lecture per.
Revise notes पढ़ मैं कैसे लेकिन P block भी करवाना है ना? ठीक है, वो मैं बता दूंगा. We'll do one thing, half of the time P block कर लेंगे, half of the time revision कर लेंगे coordination compounds का. ठीक है?
ठीक है.
रिविजन. ठीक. वीकेंड्स में कहाँ क्लास होता है? सैटरडे या डिपेंड समटाइम्स Saturday you have both sometimes maths and physics both. हाँ वो रिवीजन वो सर को बोलना, आशीष सर को बोलना. Normally he keeps this classes. Depends on his mood. Moiety means maybe the sugar moiety ऐसे लिखा है ना, phosphate moiety. पूरा पूरा पढ़ो तो. हाँ मतलब phosphate moiety मतलब phosphate group. 3 बार बोलना पड़ा. Good question. I had the same question when I was at first. So on the next class, I'm going to revise coordination. Okay, whenever we have the next class. Revise coordination. Amines भी ठीक है. Amines का पहले coordination revise कर लेंगे. ठीक है. After that amines.
So can you do aldehyde before amine?
No, like Moesha asked me first.
Okay, so it's
Then I will do aldehydes.
okay.
Yeah. On change of pH. Yeah. That's denaturation of proteins. Remember, when you, for example, the egg, when you heat it, the same thing happens. but the primary structure remains intact. Primary structure is the amino acid, alpha amino acid कौन सा है वो तो change नहीं होगा. Done with the summary. Then start doing the NCERT exercise. Yeah. We did vitamins. We did. I told you to read that. So what will I teach? Like vitamin A कहाँ मिलता है? नहीं खाने से क्या होगा? What will I teach? Then you need to learn. Vitamin B12 कहाँ मिलता है? Vitamin C. Yeah. They can. Both are interrelated. Because biology it is composed of molecules, organic molecules. Good point. So, what are monosaccharides? Dash. Carbohydrates which cannot be broken down. And you do into simpler units, SLA. Good. Ribose, monosaccharide. See, the ribose is there in DNA, right? Ribose is there in DNA. So that's a monosaccharide, right? Monosaccharide sugar. Reducing sugars. Yeah, what are reducing sugars? Positive tolerance and Felix test. And they have a free aldehyde or a ketone group. Right? Yeah, I told you a statement. All monosaccharides are dash sugars, reducing sugars. All disaccharides are dash sugars. You cannot generalize. Some of the disaccharides are reducing, some are not reducing. Online guys, send the summary of this chapter now. Send it to me. Why is your video off, everyone? Send the summary, whatever you have written. Starch is found in plants. Glycogen is found in animals. Starch consists of amylose and amylopectin. Glycogen is similar to amylopectin but more branched. Remember what was amylose? Starch के दो components होते हैं ना, amylose and amylopectin. Amylose में क्या होता है? There is no branching. Amylopectin में branching होता है. And glycogen is animal starch. That means it's something similar to glycogen, but it is highly branched. Yeah. Yeah. So how do you explain that? So you can draw the structure that one is 3 dash to 5 dash, one strand is 3 dash to 5 dash, another strand is 5 dash to 3 dash. Then you can show कि उसका adenine और इसका thymine, इसका thymine और उसका adenine. बॉन्ड वो भी पॉइंट पे लिख सकते हैं. That also you can write. Okay. That point you have to mention, okay, कि guanine और cytosine के बीच bond बन सकता है, guanine और adenine के बीच नहीं हो सकता. That point is must. Adenine and thymine can form bonds, two hydrogen bonds. Similarly, guanine and cytosine can form three hydrogen bonds. ठीक है? That reacts with both acid and base.